Thanks everyone. So I'm a master's student in the Hub, supervised by Marta and Jacob. And just following on from some of the things that Jacob was talking about earlier, I'm going to talk about how we're using the 7T MRI at the university to its full capacity to, in my study, look at the representation of sensory uncertainty in the brain. So to understand what I mean by sensory uncertainty, I want you to make a judgment now about how far away you are in your seats from me up here. So you might have an estimate that comes to mind. For example, depending on where you're sitting, you might think I'm 10 meters away. But you're also probably aware that your estimate is unlikely to be exactly correct. So you have some uncertainty in your estimate. So this might manifest as these kinds of error bars around your estimate of a couple of meters either way. Now imagine that I turned off the lights in the room. Your estimate would probably be more uncertain, so you might have these wider error bars. So over the last couple of decades, a plethora of behavioral studies have shown that humans seem to be aware of the uncertainty associated with their sensory information, and they use this knowledge about uncertainty to make better decisions. What isn't clear, however, is how this uncertainty information is represented in the brain. So a couple of really exciting studies have been done so far that have begun looking at this. And basically what they've been able to show is that uncertainty information seems to be present at the earliest stages of cortical sensory processing. So in one study by Van Bergen and colleagues, they showed individuals these oriented Gabor stimuli, and then after a few seconds asked the participants to rotate a bar to match the orientation of the Gabor that they were shown. So what they were able to show was that using a novel model-based decoding algorithm, they could generate estimates of uncertainty on a trial-by-trial basis. And basically they showed that when the representation was decoded to be more uncertain, individuals made greater errors when trying to recreate the orientation of the Gabor. So this is really exciting, as it suggests that uncertainty information relevant to behavior is present at these early sensory stages. What it doesn't tell us, however, is where this uncertainty information comes from, specifically whether it comes from bottom-up or top-down processes. So a bottom-up account might suggest that there's noise even at the earliest stages of sensory processing. So for example, in the visual system, that the photoreceptors in the retina are somewhat noisy, and that at some points in time, there'll simply be more noise in the system. So the bottom-up account suggests that at these times where there's more noise in the system, then our sensory representations will be more uncertain. More recently, however, others have suggested that this uncertainty information primarily comes from fluctuations in top-down signals. For example, if you can imagine when your attention's more focused, your sensory representation might be more precise. However, when your attention is divided or less focused, your representation might be more uncertain. So basically, I want to consider where this uncertainty information, which has already been found in primary visual cortex, whether it originates from bottom-up or top-down processes. So the technique I'm using to look at this, as Jacob alluded to earlier, is called layer fMRI. So basically, we can divide the gray matter in the cortex into 3 bins of layers: the superficial, the middle, and the deep. And we can use our knowledge about anatomy to infer where the activity comes from. So basically we know that bottom-up signals primarily terminate in the middle cortical layers, whereas top-down signals primarily terminate in the superficial and the deep layers. So using this knowledge, layer fMRI studies infer whether activity in a specific region, for example in primary visual cortex, primarily comes from top-down or bottom-up signals by which layer is more active or contains the most information. So for my study in particular, I want to consider which layer contains the uncertainty information, which we already know to exist in primary visual cortex, and use that to infer whether uncertainty information primarily comes from top-down or bottom-up processes. So here are some more pictures of some layers from a pilot subject. We're currently almost finished data collection for the project, so some exciting results coming soon. Thank you to everyone involved in my lab.
Yeah, very interesting project. It's very clear how you can use 7T to show the structural layers, but in terms of fMRI, it depends on the blood vessels. And are they distributed and at the finest level in a way that makes the layer distinction very straightforward in terms of bold signal, which is the inference?
So basically, functionally, we know that you can distinguish activity. You can get signal that's specific enough to be inferred for a layer, but how we actually define the layers is on our anatomical image, and then we get a registration from our anatomical onto our functional and register them on. So you can't visually see your layers on the functional image, but we do know that you can get the specificity to see differences in signal between the different layers.
What I'll be talking about today is a project from my PhD, which will be discussing how information processing can underlie altered perceptual experiences, such as hallucinations, which are seen in false percepts, and delusions, which can be considered false concepts. And I'm particularly interested in how this differs a lot across the continuum of psychosis, so particularly in psychotic-like experiences and individual differences in perception. So Bayesian inference is a framework where perception can be considered a weighted combination of prior beliefs or expectations and incoming sensory evidence. And this is weighted accordingly according to the reliability of information, similar to what Josh described in his example before. And the brain creates an internal model to optimize perception and influence the way in which behavior is updated. So impairments in the relative weighting impairments in the relative weighting associated with prior and sensory information are thought to reflect changes to one's perceptual experience, contributing to an altered internal worldview. So this slide's just an example showing a balanced weighting of prior to likelihood information, with the posterior, which is the purple line, representing the combined perceptual experience. So early accounts of psychosis formation suggest that an over-reliance on sensory information confounds perceptual experiences. So this is where unexpected events are more likely to motivate explanations for belief updating about the environment. So this is often seen in hallucinations as a direct experience of this internal representation, and in delusions as a cognitive effort to make sense of the experience. But there's some opposing theories that suggest that psychosis is driven by an over-reliance on prior expectations. So this is where the distinction between one's internal model and the reality of radical perception becomes less clear. So again, this is seen in hallucination formation in terms of perception in the absence of corresponding stimuli, and in the maintenance of chronic delusional beliefs, which are resistant to change despite contradictory evidence. So a lot of research has gone into clinical presentations of this, but it's unclear whether these aberrancies extend into the non-clinical continuum of psychosis in terms of people's psychotic-like experiences. So my study was to adjudicate between these two competing theories, and also it's unclear how one's subjective perception of how reliable or uncertain each piece of information is, so or refer to that as the precision, contributes to this altered information integration. So we used a behavioural perceptual decision-making task that orthogonally manipulated likelihood information from trial to trial and prior information across blocks. So the optimal behaviour was to integrate these two pieces of information depending on the reliability in each trial. And we used Bayesian modelling to compute the relative weighting associated with prior and likelihood information, and also the subjective uncertainty associated with each. We correlated these parameters with psychotic-like experiences, which were measured with the positive dimension of the CAPE, which is a questionnaire that looks into alterations in thought and sensory perception. And we collected this over 2 independent samples. We had over 1,000 participants for the whole data set. So what we found, quite briefly, was across both data sets, we found psychotic-like experiences were positively associated with an overweighting of sensory information relative to prior expectations, and that psychotic-like experiences were also positively associated with a subjective uncertainty in prior information. So this is the decreased precision in the blue on the slide there. So this suggests that the overweighting in sensory information, because the prior information is more uncertain or more subjectively uncertain than what it actually is. But interestingly, in this study, we also found that psychotic-like experiences were positively associated with subjective uncertainty in sensory information. So this meant that participants were encoding sensory information with greater noise, and there seemed to be this general overestimation of uncertainty across the task. So this meant that participants were perceiving greater instability or uncertainty in their inferred internal representation of the world in general. And this decreased precision in likelihood and prior information has been used to co-explain different behaviours that are seen in schizophrenia. For example, people completely ignoring new information and preserving on previous responses, but also heightened behavioural switching. So when they do update their behaviour, they tend to do so completely. So it just suggests that this phenomenon might extend across the continuum into non-clinical presentations as well. So just to sum up, this lends empirical support to weaker bottom-up, but also weaker top-down perceptual processes, and suggests that this might extend into belief updating along the non-clinical continuum of psychosis. And yeah, thanks to my lab and my supervisors for their support.
You were talking about sensory information. Was it across any domain, or is there any specificity to how you measured it?
So in our paradigm, we were looking at visual sensory information. So there were splashes on the screen that were more or less uncertain to denote a target. And yeah, this is generally studied across the visual domain. Yeah.
My name is Violet. I'm one of the PhD students in the Hub, supervised by Stefan and Daniel. I see you there. Hello. And my project explores the neural representations of food attributes such as health and taste, and how they may relate to aspects of food choice. So in case you missed Daniel's excellent explanation of multivariate pattern analysis, or MVPA, early on, if you joined us after lunch, I will be briefly going over it. So it's believed that the brain encodes neural representations as patterns of activity across distributed sets of neurons. A neural representation is a pattern of neural activity that stands for some environmental feature, such as your perception of how tasty a food item is. EEG, or electroencephalography, provides population measures of neural activity with millisecond precision that allows us to study the temporal dynamics of these cognitive processes in a non-invasive way. Because MVPA is really useful for looking at the spatiotemporal patterns across the distributed EEG signal, it's thought that we can access the underlying neural representations using this technique. So MVPA involves two broad steps: data collection and pattern classification. Firstly, we collect the neural data under multiple distinct experimental conditions. The data is then split into the training set and the test data set for each experimental condition. A machine learning classifier is then trained using the training data set to distinguish between patterns of neural activity associated with the different conditions. For example, viewing foods that differ in their tastiness. The classifier's performance is then tested using the test data set to determine whether the different conditions, in this case the taste ratings, can be decoded from the neural data by the classifier at a level above chance. So MVPA has been used to decode simple and abstract features from EEG data in the past, and importantly, a recent study from our lab successfully decoded health and taste representations during dietary decisions. In this study, participants rated pictures of food items on health, taste, and decision to eat on a continuous sliding scale while their EEG activity was recorded. Using MVPA, it was found that health, taste, and decision to eat information could be decoded from early EEG activity as indicated by the pink bars here. So far, I've referred to studies having successfully decoded certain neural representations. There is a common implicit assumption that if information can be successfully decoded from patterns of neural activity, then this provides strong evidence about what information those patterns represent. But even when decoding is successful, it is possible that the decoder might just be using some extraneous information that is merely correlated to the representations of interest to decode the conditions, and therefore this makes it quite difficult right now to infer neural information directly from decoder information. So how do we bridge this gap? If, for instance, the decoded information was found to be meaningfully associated with relevant behavioral outcomes, this might provide evidence towards the idea that the decoded information is indeed accessible to and maybe even used by the brain. and what aspects of decoded representations might be meaningful, we can look at decoding accuracy as a proxy measure of the strength of the underlying neural representation, and maybe decoding onset as a measure of the time at which these representations become available. There is substantial variation in both measures across individuals, and this may, at least in part, be explained by differences in the strength or time course of the underlying neural representations. To test this hypothesis, we had participants come in and complete a food categorization task in which they categorized food items as yes or no in terms of healthiness, tastiness, and decision to eat as quickly as possible. Response time was recorded, and crucially, EEG was recorded throughout. Participants also gave precise continuous ratings of each food on the same food attributes, and MVPA using support vector machines for the binary categorizations and support vector regressions for the continuous ratings was performed to decode health, taste, and decision to eat information from early EEG data when participants were first presented with the food image in the categorization task. All right, so if you squint, there will be some pink on the screen. That's very important. I'm sorry that with the projector it's not super clear here, but we did find that consistent with the previous study, there is information present within the first second of viewing the food image for these representations. The left plot shows the decoding accuracy over time when decoding binary yes-no categorizations for health, and the right plot shows the accuracy when decoding continuous health ratings. Time 0 here indicates when the food image was presented, and the pink bars, which you can see if you squint, indicate the times at which decoding performance exceeded chance levels after correcting for multiple comparisons. So around 150 milliseconds onwards, it's all pink. You just have to take my word for it. We do see similar results for taste, shown to be decodable from around the same time frame, and also for decision to eat, also decodable from around the same time. Our findings are in line with and also add to the findings from the Schubert et al. Study. It's interesting to see that we can decode these binary categorizations as well as the more fine-grained continuous ratings of the same food attribute representations. So where to from here? The next step will be to link these neural decoding measures to aspects of food choice behavior. As to which aspects, we have a few candidates. We could look at the categorization RTs from the food categorization task and see whether individuals' decoding accuracy for a certain food attribute representation might be linked to the speed at which they categorize the foods on that attribute, for example. We also had participants complete a food go, no-go task using the same food stimuli, in which participants' own ratings were used to determine the go and no-go foods. Versions of this task have been used to previously measure food-related inhibitory abilities, which might also be linked to the decoding measures. We also obtained a measure of the extent to which participants are driven by the healthiness or the tastiness of the foods when making food choices through a paired food choice task. And lastly, we had questionnaire measures of participants' dietary style and food-related attitudes. If the decoding measures were found to be correlated to any of these aspects of food choice behavior, this would lend support for the idea that the decodable food attribute information in the EEG data are indeed maybe used in a meaningful way by our brain. So to conclude, the broad aim of the project is to use decoding measures to learn more about the neural underpinnings of dietary decisions and also neural representations more generally, decoded representations more generally, as well as to develop neuroimaging tools in the lab that might be useful for more applied research in health and diet. Thank you for listening.
I missed the trial structure. In that first second after they see the image when you're decoding it, do they keep their eyes and hands still so that you're not getting like interference from the response side that they're reaching to or something?
Yeah, that's correct. So when they are viewing the food image, they haven't yet been asked the question of whether they're supposed to judge it on healthiness, tastiness, or decision to eat, and they're just completely still and hopefully fixating on just the image.
Hey everyone, I'm Lauren, and today I'm going to give you a little glimpse into some of the projects that I'm working on that broadly aim to examine the neural mechanisms of voluntary decisions and, more importantly, subsequent changes of mind. So voluntary decisions are these free, willful decisions that we make, and we typically have to have some sort of conscious deliberation between our choice alternatives and the decisional control to execute those decisions. Now, we also tend to have this accompanying sense of agency or this feeling of control over our decisions, and also quite a clear understanding of how these decisions then influence the external environment around us. Voluntary decisions are primarily driven by what we call internal evidence, which includes things like your personal preferences, memories, and goals. But sometimes it's quite helpful to also integrate some external evidence, such as your anticipated rewards and costs, and also like sensory cues like color and motion, especially when they can give you some contextual information or guidance on how to act. Now, very, very briefly, voluntary decisions are thought to be first represented in prefrontal regions, where things like the generation of intentions take place. Subsequently, we see activation in pre-supplementary motor areas, or SMA as well, where the preparation of motor activity takes place. And lastly, as we're about to execute our decision, the primary motor cortex is then activated, and the neural signals leave for the spinal cord, and the muscles used to, well, execute that decision. Another key thing is that voluntary decisions are typically encoded in medial regions of the brain, in contrast to potentially lateral regions, which are sometimes found in stimulus-driven decisions, like your perceptual decisions. The problem at the moment is that a lot of these studies were currently based on single decisional processes that kind of end once you make a decision. But what we don't quite know is how neural representations may be the same or potentially change beyond the point where you make your initial choice. And this is really important in complex and changing environments where, for example, you might change your mind about certain things. So, for example, you are, I don't know, in Brunswick, if you live in Brunswick, we're a very famous bakery there with really long line, and you're there to get their famous chocolate croissant. But by the time you reach the front of the queue, you realize it's all sold out, so you don't have much of a choice here but to get a different croissant instead. Or alternatively, what if a baker comes out and puts this this range of like freshly baked goods in front of you? Maybe that entices you to like change your mind and pick something else. Or maybe if you're really committed to your chocolate croissant, you could also just stick with it. So I think this is what makes voluntary changes of mind really interesting, in that you can integrate this additional new evidence, but you can choose to proceed either way. You could change your mind, or you could stick with your initial choice. So this is quite different from other types of stimulus-driven decisions where you're typically correcting an incorrect response, or you're spoiling a perfectly good one. So this is something that I wanted to explore a little bit more in depth and understand the neural representations of how voluntary decisions and subsequent changes of mind are represented in the brain over time, and more importantly, do they differ? So what we did was we collected some surface EEG recordings and also some stereotactic EEG recordings. So SEEG, as was mentioned earlier, it's typically only used in epilepsy patients to determine the seizure onset zone. So SEEG uses, never mind, there's no mouse, but if you can see on the extreme right, we see this really thin, long lead with some electrode contacts spaced across it, approximately 18 of them. And then they're implanted into the deep cortical structures of the brain. So if you can see in the middle photo, each line or each colored line represents a different lead, and it's spaced out across the brain. So with SEEG, we get a little bit more spatial resolution on top of the amazing temporal specificity of both modalities. So in our task, it was quite simple. As you can see here, this is my representation of what Daniel looks like, who's wearing the same color. So the task required participants to just collect the most colorful combinations of balloons by the end of the experiment. And we had 4 different conditions. So we had a free condition where we showed 2 options and participants could pick whichever color they wanted. We had a forced one where we only showed 1 option and they were forced to pick it. We also had 2 change of mind conditions. So this is like an initially forced decision. And then after they picked that forced option, we gave them 1 additional choice. So participants were now free to either switch to the new color, or they could feel free to stay with their initial choice. And conversely, we gave them 2 options at first, and after they picked one, we then removed 1 option. So this then forced participants to either change colors, or they were forced to stick with their initial choice. So our main analyses, or one of our main analyses at least, is MVPA, and because people have already explained this in depth, all I'll say is that we're using MVPA to decode between different types of conditions or choices, for example, free and forced choices. But the one that I wanted to talk about today is contrasting or decoding between free initial choices, and also free change of mind choices, as you can see in the red boxes. So I find this particularly interesting because they're essentially or objectively kind of the same types of decisions. They're just made at different time points. One was made initially and one is a change of mind. So the question here is, are they still represented similarly or quite differently? And so with some preliminary data here that I have, we do see that we can decode between these two choices from about, oh yeah, mine's here. Anyway, so from about 100 milliseconds after the time point that participants saw their choice options. And naturally, we can do this for the forced choices as well, and we see fairly similar results. So this is based on the surface EEG recordings with 64 channels. What we want to do with the SEEG data is to run MVPA multiple times on different subgroups of electrodes. So for example, we know that the prefrontal regions are quite important in voluntary decisions, and also the parietal region. So we may use different subgroups in those areas, as we might see decoding accuracy for earlier time points in prefrontal regions, and then accuracy or higher accuracy at later time points for parietal regions. Naturally, we can also zoom in into specific areas within prefrontal regions, like such as anterior cingulate cortex, and also pre-SMA and SMA. And as I mentioned before, we do see that voluntary decisions are encoded in the medial region, so we may also group different electrodes in medial regions. And I'll just wrap up there, thanking my supervisors, my lab, and also all my collaborators, and most importantly, my participants and patients who have been really nice and engaged in this task very, very well.
Were there any sex differences in choices? I'm pretty sure that in whether people switch or stay the same.
At the end of the experiment, we did ask participants to rank their favorite colors and whether they made a lot of their choices, like whether they switched or stayed, based on their color preferences, and the answer was yes. So for that reason, if, for example, there was a free choice and people already attained their favorite color, they chose to stay with it. Or if they were kind of a bit more goal-directed and they were trying to get a colorful bunch, they just tended to stay more. Yeah.
My name's Tim. I'm supervised by Hinza Hoggendorn and William Turner, neither of which are actually at the school anymore. They're both based at QUT in Brisbane, but I'm still part of this hub. So today I'm going to talk about the extent to which shared mechanisms contribute to motion position illusions. So motion position illusions refer to this broad class of visual motion illusions where the position of an object in the context of motion is incorrectly perceived. So by in the context of motion, I mean the object could itself be in motion, it could contain motion, or it could be surrounded by motion. Now, this is quite a broad definition, so as a consequence, there's several illusions that could theoretically be considered part of this group of illusions. In this study, we were only interested in kind of the 8 most prominent illusions, which I've got up here now. I don't have time to go through all these today, but if you're interested in seeing these illusions, I've got a QR code on my last slide, which you can scan, which will show you each of the illusions. But just as an example, I'll go through the flash lag effect as an example. So this is our version of the flash lag effect. This is just an example of a single trial. So we have a rod that's rotating clockwise, and after the rod has rotated 90 degrees, we present a static rod in spatiotemporal alignment with this moving rod. So the moving rod and static rod are physically aligned, so they look something like this. But what happens is the moving rod is perceived in a position further along its motion trajectory, such that the static rod is perceived to lag behind the moving rod, even though they are physically aligned. So this is just one example of a motion position illusion, but hopefully it gives you some idea of this broader class of visual illusions. So the reason we're interested in these illusions is because it remains unclear the extent to which these illusions are caused by shared or dissociable processes. So because these illusions look phenomenologically similar, there's a tendency for researchers to assume that these illusions are caused by the same shared underlying architecture and mechanisms. And this has led to people trying to propose theories that can try and explain all these illusions. Unfortunately, there is now recent experimental evidence which seems to suggest that some of these illusions cannot be caused by a single mechanism. You need an interplay of multiple mechanisms to explain some of these illusions. And there's also additional evidence with 2 illusions, which has shown that these 2 illusions, which are typically assumed to share the underlying factorial structure, actually are not caused by the same underlying factor. So we have to be cautious when generalizing these results because they're just with one illusion with two illusions. So in this study, we basically really wanted to get the factorial structure of these illusions by having 106 participants complete these 8 illusions twice in 2 separate sessions. We used an individual differences approach. So basically with this approach, we can assume that individuals will have a similar illusory susceptibility to illusions that are caused by the same underlying factor. So on this basis, we would expect that illusions that share underlying factors would correlate with one another, and illusions that are caused by dissociable processes would not correlate with one another. So here I've got the descriptive statistics for each of these illusions shown by these marine cloud plots here. The blue colors you can see show the values associated with session 1. The red colors show the values associated with session 2. The dashed black line you see down the middle represents the point of 0 illusory effect. So anything to the right of this indicates a participant experienced the illusory effect as we would expect. Anything to the left of this indicates that participants experienced the opposite of the illusory effect as we would expect. The solid colored line shows the mean illusory effect for each session. The box plots show the median, the interquartile range, the min, and the maximum. And those dots show participants' raw scores on each session with a line connecting a participant's raw scores across both sessions. And finally, the Spearman's row values in the top right show the test-retest reliability between session 1 and 2. So when we look at all this information, we can see that all illusions had a strong illusory effect. We can see that there are individual differences in the presence of these illusions. Some participants experienced the effect as we would expect. Some participants did not experience the illusion, and some participants experienced the opposite of the illusion. Now, these individual differences aren't due to noise or some other non-perceptual factors because we observed very strong positive test-retest reliability statistics. So because of this, we averaged the illusory effect across sessions and then looked at the correlations between illusions, which gave us this correlation matrix. After Bonferroni correcting for multiple comparisons, we got 2 significant correlated clusters of illusions. These are shown by the yellow fluoro boxes. So these correlated clusters of illusions, because they correlate with one another, are likely to share underlying mechanisms. But the fact so many illusions do not correlate with one another also seems to suggest that there are dissociable factors contributing to these illusions. So our evidence seems to suggest that, firstly, a single common factor cannot explain motion position illusions. Additionally, it shows that there are dissociable mechanisms contributing to these illusions. And our results also encourage the need for the current theories, which seek to explain why people experience the effect as they would expect, to now be adapted so they can also explain individual differences and why people experience the opposite of the effect. So thank you all for listening. I also want to thank my collaborators. And yeah, if you're interested in seeing the illusions, scan that QR code. And if you want to read the publication, you can scan the QR code on the right.
Yes, thanks for the great talk. I was wondering, it looked like in session 2, the illusions, most of them get a little bit weaker. Is that right? So
yeah.
Do you think you can get rid of the illusions altogether by doing it 100 times?
Yeah, fantastic question. We have no clue why the illusory effect reduces over time, and there's a tendency just to ignore it in the literature, that it reduces. We're not sure why the effect reduces. We kind of speculated possibly some practice effects or something else going on. But I think that there is a tendency in the literature when you do repeat it weeks after weeks, you still have the effect, but it does decrease. We just don't know why. We only had 1 week between sessions, so we can't really speculate on if it would just keep decreasing the more you did it. So, yeah.
As Natalia said, I'll be looking at the cognitive mechanisms underlying perceptual change in mind decisions. This is in contrast to some of the other types of decisions that you've just heard about, such as voluntary decisions and dietary decisions, from Violet and Lauren, who are also in the same lab. And yeah, so I'm going to really dumb it down and talk about perceptual decisions as a very basic, very fundamental. And also, yeah, I'm supervised by Stefan and Daniel in the back. So to start off with, first of all, what are perceptual decisions? I probably don't need to define this for you guys. But imagine you are crossing a road. You might need to have a look at whether it's safe to cross. So you might be looking for oncoming cars, for instance. So perceptual decisions are basically predominantly based on the sensory evidence that you see in your environment. And typically these decisions are either right or wrong. So in this case, hopefully I don't need to tell you what is the right or wrong decision here. So commonly used perceptual decision-making task is the random dot motion task, So in this task, participants are asked to judge the direction of a set of moving dots, where only a fraction of the dots are moving in a coherent direction. And based on these kinds of experiments, we have these decision-making models, and they model this process, perceptual decision-making process, as a noisy sequential sampling process. So essentially what you have is a decision variable accumulating sensory information over time until it reaches one of two decision boundaries that correspond to two different decisions. There are also other models that use the same process, but instead might have multiple accumulators for each decision, and the winning accumulator dictates the decision that you choose. So in one study, where participants used a response lever to respond to the direction of a set of moving dots, they found that predominantly people were able to just move directly to the response option that they thought corresponded to that direction. However, on some trials, people would go towards one response option and then deviate towards the other response option. And this is what is defined as a change of mind. So just coming back to the drift diffusion model that I talked about earlier, the question remains, why do people change their mind if they don't actually receive any more information? So when people initiate their movement, the dot display disappears. So what is causing these change of mind decisions? So Resolage and colleagues propose that because of sensory and motor delay, so the time it takes you to process that information that you're seeing and also to respond to that information, there's actually a window of time here where you actually don't, this information in this window doesn't reach awareness until after you've made your initial decision. So essentially, this accumulation process can continue, and if that accumulation reaches a secondary change of mind bound, then it is thought that you change your mind. So this is the crux of the extended drift diffusion model, which is used to model change of mind decisions. And this is supported by the way that individuals use information in these kinds of tasks. So a feature of the random without motion task is that each sample has different amounts of evidence. So some samples have more evidence or less evidence than the mean. And if you average these residuals over the course of an experiment, you would expect this to average to 0. However, if you group or average by different types of decisions, so as an example, I put here correct and error, and correct change of mind, error change of mind, you actually get these information usage profiles that can tell you what information in the decision-making process is strongly contributing to different kinds of decisions. So using this technique, what they found was that there's actually, just prior to that initial decision that you make, a difference between the change of mind trials and all trials, indicating that this period, just before the initial decision, is critical to change of mind decision. So this is consistent with that extended drift diffusion model that I was talking about earlier. In another study, they had people make a judgment between two squares as to which square was higher in luminance, and instead of using a response lever, they used button presses to make the responses. And what they found instead, which is really interesting, is that actually the first sample of evidence, or this very early evidence, could differentiate between the different kinds of decisions, which is in direct contrast with the previous study, and also sort of begs the question as to, does early information, so the very first sample of information, contribute to change of mind decisions, or is it just this late information where when you're making that decision, you don't actually know the information, and then it comes afterwards to your awareness? And this is the aim of my PhD, my first PhD study. And instead of using the two tasks that I talked about, I'll actually be using this perceptual grading task. People hate this task, it's very difficult. Please do my study. No, I'm just kidding. But you basically make a judgment between two overlaid gratings, so towards the left or towards the right. And yeah, you have to judge which is more dominant in contrast. So here's a trial diagram. So in these kinds of studies, you show pre-decisional information, people make an initial response, and then you receive more information. and you can choose to change your mind by making the alternative response. So I'm currently collecting data for this project, so stay tuned for that. But otherwise, thank you very much for listening, and also I want to acknowledge the team. Thank you very much for listening.
What is the power in these types of effects? Do you have to test a lot of people to just determine the result, or is it more of a kind of within subject you can already observe these differences?
You do need a lot of data for these kinds of studies. At the moment, we don't actually know exactly how much. I'm basing this off of Will's study, which is the luminance judgment task. But yeah, we don't actually know exactly how much you need. We are expecting to see on an individual level, because we are collecting data over multiple sessions for each participant, to see this sort of consistency across individuals as well. But yeah, I'm not really sure at the moment.
Hi everyone, I'm Rosie, and I'm supervised by Olivia Carter and Professor Lucy Palmer over at the Florey. So psilocybin is a psychedelic compound. It's a partial agonist at an array of serotonergic receptors, but it elicits its main psychoactive effects via the 5-HT2A receptor. And the 5-HT2A receptor is most densely expressed in pyramidal neurons of the prefrontal cortex, and activation of these receptors by psilocybin alters dendritic excitability, so it alters brain activity. What I'm particularly interested in is the effects of psilocybin on the mouse dorsal cortex. So to explore this, I use a technique called wide-field calcium imaging. This technique requires the use of double transgenic mice that express the fluorescent protein GCAMP6F in layer 2-3 pyramidal neurons. So when blue light is incident on this protein, it emits fluorescence that is measured as a proxy for a change in neural activity. So using wide field, we can look at the changes in neural activity that underlie behavior. So I train my mice on different tasks, and one of the tasks that I'm training my mice on at the moment is a detection task. And within one imaging session, the mouse will complete the detection task around 100 to 120 times. And there are two types of trials within the detection task. So they've got go trials. So within a go trial, the imaging begins. There's a baseline period of 3 seconds where nothing happens, and then the animal experiences a forepaw stimulus to the right forepaw. This tells the animal to lick to receive a sucrose water reward. And if it licks correctly, it is a hit. If it doesn't lick, it's a miss. And then there's also catch trials where there's no stimulus, and so the animal must withhold from licking to get a correct rejection. If it licks in this period, it's a false alarm. So for each cohort of mice I have, which is usually an N of around 3, this is the rough timeline. So when I first get my mice, I administer tamoxifen across 3 days, and this activates the GCAMP in the brain. Then I perform a translucent skull surgery, which enables me to image through the skull. And then I habituate the animals to handling and head fixation. Then I naive image the animals. This is the first time they experience the forepaw stimulus and receive the sucrose water reward. And then I train them on, at the moment, the detection task, and maybe I will train my mice on the short-term memory task in a few months. But yeah, so the detection task takes a week to train the animals on. And then I begin imaging. So I image from day -1 to day 14 relative to day 0, which is the day of psilocybin or saline intraperitoneal injection. And I administer psilocybin at a dose of 1 mg/kg, which is high dose relative to the therapeutic dose that is being administered in clinical studies for psilocybin. So you can barely see that, actually. That's really bad. But anyway, this is a representative example of the flow of cortical activity through the dorsal cortex during the detection task. And this is an example from one animal on day -1, so the day before psilocybin or saline administration. And it's the average of all the trials that the animal responded to. So the go trials, the animal correctly responded to, so all the hits. And during the baseline period, what you're supposed to see is that there's no particular activity in any region. But during the stimulus, you see increased activity in the contralateral somatosensory forepaw region. And then during the response, you see increased activity in the frontal motor regions, so M1 and M2, when the animal is planning and executing its response. And then during the reward, you see increased activity in regions such as like the olfactory bulb, when the animal is smelling and tasting the sucrose water reward. So I still have fairly low N numbers, but this graph shows the performance of my 4 animals in the detection task, and what it appears to show is that on day 0, so the day of psilocybin and saline administration, there's a large decrease in hit rate. And what I noticed was that mice not engaging with the task on the whole, and then sort of snapping back and getting a few trials correct, and then going back to not engaging. This graph just is an example of the type of data that you can get out of wide field. This shows the change in neural activity, in this case, the contralateral somatosensory forepaw region, to the right forepaw that the animal's receiving the stimulus to. And yeah, really too early to infer anything from this data, but this is just an example of the type of thing I get out. So what are the implications of this research? Well, it will help us to delineate the effects of psilocybin on the cortical activity which underlies behaviour. It will inform us of the translational validity of the psilocybin-treated mouse model in terms of changes in brain activity. Are they similar to that which we see in the human condition? And it provides scope for future experiments, so potentially things like using G protein receptor blockers to look at the links between the biomolecular mechanisms of psilocybin and the corresponding changes in neural activity, and potentially maybe even looking at things like non-hallucinogenic psychopathogens and seeing if they cause similar changes in neural activity to psilocybin. So I just want to say thank you to Professor Lisa Palmer and Professor Olivia Carter, and to both of my lab teams, and thank you for listening.
So why was this specific task chosen to test the paradigm?
Initially, I was going to go straight to the short-term memory task, and I had 2 mice in my first semester, and tried to train them on the short-term memory task, and they just never learnt it. So that was a bit of a waste of time for me. But I've refined that task now, so hopefully the next time I attack it, they'll get it a lot quicker. So actually, Olivia suggested that I just go straight in with a more simple task, and it's really been useful to me just to understand the whole pipeline as well with the detection task.